questions, but they only gave me the front of the pages. So if you look at the front, it's through 22, and you turn over, it's question 41. I was never given questions 23 through 40, so, and then 57 through 81, 58 through 80, I wasn't given that one either, so. What I got, you guys got. Yeah, just make up your own questions and answers. So I gave you 3 different types, or 3 different sets of questions for the finals, so hopefully that'll give you a good enough broad scope of where you need to study, where you need to spend your time. I would suggest, first of all, not wait until Thursday. I know I've gone over this before, but don't wait till Thursday to take this test. Everyone else and their dogs will wait till Thursday. The testing centers will all be full. And with everybody on KaiTester, if KaiTester goes down and you didn't get to take your test, you get a 0. You don't get to remake it. You don't get it. If you're in there and the test goes down, as far as you get, that's as much credit as you get.
No.
Yes. So they've given you Tuesday. Actually, it's open now, so you could go now and take your final. So there's no excuse that you have to wait for the last day to take your test.
Does that happen a lot? So
It happened last semester. It crashed at 6 o'clock. They didn't get it back open until 8:30, so people had a half hour to finish wherever they were at. And no one else was allowed in. So yes, it does happen.
So they were just sitting there waiting.
Just sit there and wait.
you can't come, like if it crashes
Can't
like on
leave.
Tuesday.
Oh, if it crashes, it's not gonna crash on Tuesday. Oh, no. If it crashes on Tuesday, you can go Wednesday, you can go Thursday and do it. If it crashes like on Tuesday and doesn't get back up till Thursday, then they're gonna make it so you can do it another time. Because you did not have any time to get it done.
That's just if it does it Thursday.
Yeah, if it does it Thursday,
But it's not gonna crash on like on Tuesday or
most likely not, because most people aren't gonna be taking it there, so the computer's not gonna be overloaded. So,
What testing center is less crowded?
the one downstairs, you gotta check to make sure it's open for finals week though.
I've got one scheduled.
Short one. Okay. So, don't wait till Thursday. You can take it on Monday, you can take it on Saturday. Just don't wait till Thursday because you will be held accountable if you can't get it finished. All right? All right. Now we'll get into muscles. Any questions before we start?
Oh, I have a question.
Uh-huh.
What about muscular system?
Didn't know. Oh, I'm just joking.
So that there's like a paper that, is this like the paper that just says like some modules we have to study, or is there a different paper that we get for the
No,
final?
no, the finals one looks like this. If you go to Canvas, near the bottom, it'll say module, objective review for final,
Oh, okay.
and you can print it out. Like this was objective 1. On your test of 100 questions, 8 of them are gonna be on objective 1, or module 1. There'll be objectives 5, 8, 9, 10, and 11. That's it.
Okay, so that's just, we just get on Canvas and.
Yeah, go to Canvas, go down to the end, it should say under finals, it should say objectives review for finals. What I would do is I would go through, take those objectives out and put it in a separate binder and study from that binder, or the ones that you know, just flip through them. Don't take a lot of time. The times that the ones that you kind of remember have to go back and remember. Take a little more time on those. But you should, like these ones, give an example of each level of organization of the human body. Arrange the levels in the correct order from smallest to largest. What are the levels of the organization of the body? Chemical, cellular, tissue, organ, system, organism, CC-TOSO. Okay, so give an example. What's chemical? Calcium. What's cellular? Squamous, cuboidal, amoebas, cellular level. Tissue, connective tissue, epithelial tissue, all the different things, okay? Organs, heart, lungs, hope you can get that. Systems, urinary system, digestive system, blah, blah, blah. And an organism, hopefully an elephant. You can figure out that's an elephant. Okay, so that's all it is. I mean, you might have to review just a little bit, but go through and do that. Put those objectives and only study those objectives, because those are the only ones that will be on the test. All right, muscles. Think. You ready?
No.
No?
Just as long.
It's gonna be, this chapter is long? It's not that bad. There's a learning part and then there's the memorizing part. The first 2/3 is learning, and then after that it's all just memorizing. Okay, so we're gonna try and get through the learning part, and then you guys can memorize muscles on your own.
So what objective is that going to?
About 30, 29.
That's what we're usually
Yeah, we're gonna try and get through that, and then through the rest, which is 36, 35, 30 through 35, that's just memorizing muscles, origins, and insertions and actions. Okay, so what are the primary functions of the muscular system? Heat, movement, stabilization,
storing, moving, substances.
okay. What do you store in muscles? Calcium, what else? Not as much. There's one really big one.
Digestive.
Um,
Lymph fluid.
no, it's also stored in the liver. We talked about it. It's a lot of glucose strung together. Glycogen. You store glycogen because glycogen is broken down easily into glucose, which then can be converted into ATP. Muscles need ATP. Yeah. Okay. So yeah, that's just, so know those 4 functions. It's not too hard. Also, we talked a little bit about this last module, 2 modules ago. When we compared and contrast 3 types of skeletal muscles: cardiac, smooth, skeletal. Okay, you're gonna have to rehash that again and go over which one is branched, which one is single nucleated, tapered on the ends, as it is smooth striated.
No.
Why not? What striates skeletal muscle and cardiac muscle? Sarcomeres. Does smooth muscle have sarcomeres? No, it doesn't. It has it arranged in, not, instead of being stacked on top of each other, how's it arranged? Like what? Like a mesh vegetable bag. Exactly. You've seen those vegetable bags on page 435. Okay, what it does is it has them arranged in a mesh-like way, in which so when they contract, it squeezes the muscle into like a bulb versus a tapered diamond. Okay, that's important to understand, important to know that smooth muscle does not have sarcomeres. It has actin and myosin. It just doesn't have a sarcomere. Okay. What makes a muscle unique? Let's say that. A muscle cell unique.
That has the nucleus, the nucleus on the outside.
Okay, that's a skeletal muscle. And why is that?
Not sure.
What does the word succinctum mean? As multinucleated, but it's, why is it multinucleated? It's fused. What's fused? A bunch of cells fused together to string out a long fiber. Okay? That's the synctum, is when you put a bunch of cells together to make a long fiber. Okay? The skeletal muscle, do you know how long the longest, well, I should say it this way, how long is a muscle fiber? As long as it needs to be, but how would you define it though?
It goes from the beginning to the end.
Of what?
Muscle.
Of the muscle, exactly. So my bicep muscle goes from my shoulder, one fiber will run all the way to the elbow. That's one fiber. Then you have a bunch of fibers together that makes up the entire muscle. One fiber runs the length of the muscle. What's the longest muscle in the body? What's that?
I don't know the name of it.
Somewhere in the back.
Yeah, it's the one that.
Starts here on the hip,
It's,
goes over to the inside of the knee. Starts with an S.
semi, no.
Let's see where it's labeled for you. They're not labeled for you?
I remember reading it.
It's the sartorius.
Where's that at?
I'm not seeing it on the leg there. It's there, but it's actually blacked out. It's right above the rectus femoris on page 477, but they widened it out. But you can see it starts at the hip and goes all the way to the inside of the knee. So for mine, mine's about 2.5 feet long. That one muscle fiber is 2.5 feet long. Okay? That's why it's multinucleated, because you have a whole bunch of cells being put together, being fused together. What do we call those fuses in the cardiac cell? Those fusion points. The discs, the intercalated discs, exactly. Those are the fusion points for cardiac cells. Okay. What else is in cardiac cells that make them unique?
They're branched, gap.
Branched and gap junctions. Exactly. So you've got gap junctions. What are the purpose of gap junctions? Communication. Exactly. Okay. Able to transport, and also desmosomes. Why are desmosomes important?
Hold it together. Spot welds.
Spot welds hold together. Do you think the cardiac muscle has a lot of stress on it? Do you want rips and tears in the heart? Can you deal with a rip or tear in your arm? It's painful, but you can deal with it, right? You're not going to die. What happens if your heart starts ripping apart? Yeah, that's not so good. Yeah, that will not be any good. So know those different function or different characteristics of each that make them unique and kind of what has what. Okay. Now I said that one muscle fiber runs the length, and you have a lot of muscle fibers. The largest that you actually can see we call what? Grouping. What is this called?
The skin.
What's under the skin? Muscles. Which one are we talking about? Which objective? Or
Skeletal.
well,
Bicep muscle?
we're talking about this module. We're talking about muscles. Muscle is the gross anatomy. You can actually see the muscle. Okay. Muscle, is that all it is, or is it made up of smaller things? Smaller things. What's just smaller than the muscle itself? It has a bunch of bundles. Muscle is made up of a bunch of bundles of fascicles. Okay, those fascicles, are they the smallest unit? No, they're made up of a bunch of muscle fibers, which we also call muscle cells. Okay, Muscle cell, muscle fiber is the same word, okay? Muscle fibers are made up of myofibrils, which are made up of sarcomeres. What's the smallest functional unit of the muscle? Sarcomeres. Sarcomeres are actually made up of what though? Filaments. Which ones? Thin and thick, known as actin myosin, and what other two proteins? Well, actually, there's a few other, but what are the two major ones that are associated with the actin? Tropin, troponin, and tropomyosin. Exactly, very good. Okay, so the actin is just not actin. There's troponin and tropomyosin that also work with it. So we'll talk about that in a minute. But so sarcomere is the smallest. You get a bunch of sarcomeres, and that makes up a what? Myofibril. You put a bunch of myofibrils together, and you get a muscle cell. You put a bunch of muscle cells together, and you get a fascicle. You put a bunch of fascicles together, you actually get the muscle itself. Okay. How is that all held together? Different layers of connective tissue. What goes around the outside muscle to hold everything together? Epimysium. Okay, what's that made out of? Dense, irregular connective tissue. Okay. What about the perimysium? Where's that found? Around the fascicles. How many of you have had pulled pork sandwich or rump roast or something like that you pulled apart? It comes apart in little strings. What are you pulling apart? The different memories, but you're actually pulling apart the fascicles. The ones that you can see, that's the actual fascicles that you're pulling apart. Didn't bother you up till now.
By the time we finish this class, I'm gonna be vegetarian.
Nah, you'll learn in the next module that how important meat is for us. Chicken. Yeah, just don't eat Jell-O. I like Jell-O. Didn't bother me a bit. Okay, so know where these different. Oh, sorry. What's the endomysium around? Each muscle fiber, muscle cell. Okay. What is the name of the muscle cell membrane?
Sarcolemma.
The sarcolemma. Exactly. The sarcolemma is also it's the same way of saying the plasma membrane of the muscle cell. Okay. The muscle cell has a plasma membrane on it, what we call sarcolemma. Did Richardson call it a plasmalemma when we were talking about regular tissue cells, their plasma membrane? No. You could also call this plasma membrane a plasmalemma. Okay, it's the same thing. Lemma just means membrane. So it's a plasmalemma or a sarcolemma because it's inside the muscle. Okay. That picture, I think is in that handout I gave you, that bottom picture on 437. It would be wise to be able to put all those lines in. It's kind of hard to see, but I think it's in your book. I think it's the second page. Just,
All of them?
I'll go back to it.
It's good in the book.
Yes, it's in the book. It's one more. There you go. It's the second page, right? It's in the book. It's much larger. What page is it in the book? 304.
Or 306. Well, how far up do you need to know it?
All the way to muscle and tendon and bone.
So from bone to there, then it's on 304.
304. What's on 306? Yeah, you need to know those ones too. So 304 and 306 are good pages to know in your book. What pages? Sorry.
Right there.
304 and 306. Because you will have to identify different layers and levels of that. What I've also put in that handout is the breakdown of fascicles, breakdown of myofibrils, and the breakdown of sarcolemmas. Okay, so be able to label those also. Those ones I think are also in your book. It's where the original pictures came from. You will, if not have to be able to label them, you will definitely have to be able to give them in question form. What is a myofibril made out of?
You're supposed to tell us.
Question mark.
Say those sometimes.
Sarcomeres, exactly. Okay, so myofibril is made out of sarcomeres. So you can have it that way written in a sentence, or you might have to label one of these pictures. Okay, this is going to be a big part of knowing on the test. The myofibril is the first level that we see what? The actual workings of the functional units. What are the two main parts in a myofibril that allow the muscle to function? No, that's in the sarcomere.
T-tubes and the sarcoplasmic reticulum.
Exactly, T-tubules and the sarcoplasmic reticulum. Okay. What are those two things together called? Triad. Okay. You have one T-tubule to two SRs, sarcoplasmic reticulums. Okay. What? Well, a myofibril is what? We just said this.
Sarcomeres.
A bunch of sarcomeres. Okay, so if you have a bunch of them, is a myofibril flat, or is it round and thick? Well, perspectively of where you're at, very small, but I mean, in regards to how much is inside. Okay, if the sarcolemma goes around the entire muscle cell, are there myofibrils that don't touch the sarcolemma?
Yes.
Yes, because the sarcolemma makes up goes around the muscle fiber. What makes up a muscle fiber? Bunches of myofibrils make up a muscle fiber. Okay, so you got bunches of them. Does every single one of them touch the outside? You've got to get a way to get the information that's going along the outside of the muscle fiber all the way down inside to those bundles that don't touch the outside. That's called the T-tubules. Okay, so the T-tubules run deep across the myofibril so that you can.
So each myofibril doesn't have like a membrane per se?
No. It's well.
What layers can you have the...
So what you have is, no, they don't have their own plasma membrane or sarcolemma. The sarcolemma is around the muscle fiber, and then inside the muscle fiber you have a bunch of myofibrils. Okay, the T-tubules start out here and they run down and go across. Okay, allowing the signal that runs down, which we'll talk about in a second, which is an action potential, to go in here and then to go through all of those myofibrils and release calcium into the sarcomere. Because if you pull one of these out, what's in here? A bunch of sarcomeres. Okay, and we'll talk about why it needs to be stimulated that way, but know that the myofibril is the first time we start seeing T-tubules and sarcoplasmic reticulums. Okay, does that make sense? So a sarcomere is what, by definition? Z-disc to Z-disc. It's also called the what of the muscle? functional unit. It's what contracts to allow a muscle to contract. Okay, so a sarcomere is the functional unit. And sarcomeres are from Z disc to Z disc. Okay, you're gonna have a whole bunch of these lined up right next to each other, right? The thin filament is what? Actin. The thick filament. Myosin. They're not perfectly lined up, but. Okay, so this is one sarcomere, another sarcomere, another sarcomere, another sarcomere, right? So how does these become functional units?
Like when the calcium comes in and it attaches to the myosin and twists.
All right, but how does the sarcomere make a muscle contract? I understand all those parts, but they move over, and what happens to the Z line? What happens to the sarcomere?
And they overlap.
They shrink. So this is a relaxed state. This would be in a contracted state. So the Z disc to Z disc, or the Z line to Z line, come closer together. That's how a muscle contracts. Okay, we'll talk about how it actually contracts with the sliding over each other, but that's how. So you get this one to contract, and this one contracts, and this one contracts. What happens to the overall length of the muscle? It contracts. Does the muscle fiber get any shorter? No. It just contracts. What happens when you flex the muscle? It bulges, right? Because the muscle fiber didn't get any shorter, but you put it in a smaller space because all the sarcomeres came together. Does that make sense? So the muscle never gets shorter, it just, the inside kind of contracts itself. We all good? Okay. So what are the different sections of a sarcomere? What do we call this area right here? No.
The eye.
I band. You can think of this one as only actin in it. There is no myosin in it. Okay? What do we call this one that has both actin and myosin? A band. This has actin and myosin. And then we have one that is just myosin. The H zone. You have to be able to label these so that if they say, what is this? What's in the A band? What's in the I band? Where's the H zone? You okay with that? So I band is actin only. The A band is both actin and myosin, and the H zone is only myosin. Okay. This is important to understand is the I band lighter or darker than the A band? The I band versus the A band is gonna be lighter, because what's in the I band? What's in the A band? Actin and myosin. Okay, so you have a light band and then a dark band, a light band and a dark band. Does that sound familiar? What does that do? Striations. This is what happens, this is how you get striations in skeletal muscle and in cardiac muscle, is these sarcomeres lined up right next to each other, the A band and the I band's lined up. So you get a thick and a thin looking layer. Okay, does that make sense? Because if you actually look at a muscle cell, the skeletal muscle cell, which way, go back to 432, which way are the striations going? They're going across the fiber, okay? If they go across the fiber, which way is so this is the fiber length, the striations are going across. So it's not the actin and the myosin making the thick and thin, it's the I band and the A band. Does that make sense? All right, so we've talked about the functional unit. How do we signal the functional unit? This is, everyone's been okay so far. This right here, from here until I say stop, is the real part, the meat that you have to get out of this. Okay, there's 9 steps, and it's how a muscle contracts. Okay. Just a little pre-warning here. So we'll take as long as we need to get to where you guys understand it.
This is the meat of the test too.
A good portion of it. Yep, a really good portion of it. You're gonna have a little bit of that anatomy thrown in. You have anatomy at the end thrown in. You have levers. You're gonna have how we name muscles. But yeah, this is a good portion of the test. Okay. We get a signal from our brain that runs down a what? Neurons. So we have a neuron that's going to run all the way from the brain down to the muscle that we want to move, right? Okay. That muscle is going to stop at something called a what? Neuromuscular junction. Does the neuron actually touch the muscle? No. What is that space between the neuron I got all these fancy colors, I'm going to use them. Between the neuron and what is this? What would this be? Sarcolemma. So what layer of the muscle are we at? Muscle fiber, exactly. So the neuron comes down past the muscle, past the fascicle, to the muscle fiber where it innervates. Innervate is what we're going to call electrical current. We'll talk about this. So this is the sarcolemma. We'll talk about these in a minute. Okay, so we have a signal. What form is that signal in coming down the neuron?
Calcium.
Now let's look at what is it? Heat? Is it electrical? Is it chemical? Is it movement? Is it mechanical? Electrical. And what do we call that? Action potential. So you have your AP, action potential, which is an electrical current that comes down, gets down to here, and what happens? What's inside here? Yep. A bunch of little vesicles, and we'll learn next module how it goes from there to here, but just know for now it goes from there to here, and we'll expand next semester how that does it. But these come down and release through what type of exocytosis? They release all their junk into the cleft, right? Then what happens?
And the channel opens into a sarcolemma.
Okay, there is a receptor here that is specific to this neurotransmitter, acetylcholine. So it goes in it, and we're gonna, again, tip the iceberg, create a new action potential. Okay. Next semester, we have 3 modules, 11, 12, and 13, on explaining in depth how this happens. For right now, you just need to know it goes from electrical to chemical to electrical. What do we call that? Changing from one form of signal to another? What did you say? Transduction, very good. Did you say transduction? Translation, very close though, very close. Actually, I was like, well, I'm proud of you, way to go. Yes, transduction. Yeah, that was cool. I remember too, that was actually very well done. So transduction is going from electrical to chemical to electrical. That'll be on your final, if not on this module. So remember that, okay? So you have this action potential, and what's it gonna do? There's a theory that you were taught. If you get an action potential, what's going to happen to it? All the way. It's what theory? The all or none theory. In other words, if you create this action potential, it's going to go all the way down the entire length of that muscle fiber. There's no stopping it. Once it starts, it's going to go. It's an all or nothing. That's what that theory says. If it starts, you got it. If it doesn't start, nothing. Okay? So that's the all or nothing theory. The electrical current, the action potential will run down the length of the muscle fiber. In the myocardiogram, what part of that myocardiogram is that? On page 456. What do we call the time it takes for that muscle fiber to get the action potential all the way down the end? The latent period. Okay, so it takes, and how long does it take? About 5 milliseconds. A little fast, huh? It runs pretty quickly. Okay, so it's going to run down the entire length of the muscle. Okay, it's also going to go down these T-tubules, which go where? Inside the, in between the myofibrils, not micro, myofibrils, muscle myo. Okay, I said that all the time, and Richardson got rolled out of me. It's not micro. Get micro out of your head. Myofibrils. Exactly, but I'm glad you got it. Okay, so myofibrils, they go in between the myofibrils. And what do we say was on either side of them? Sarcoplasmic reticulum. What's inside a sarcoplasmic reticulum? Calcium. When that signal comes through here, what is it going to do to these sarcoplasmic reticulums? There's actually what's called a voltage-gated channel. You don't need to know this. But when the bolts come down, it opens up that channel. And calcium is going to flood out. Why? It's at a high concentration gradient. It's going to go to a low concentration gradient. What do we call that? Diffusion. Exactly. Is that active or simple transportation, or what I'm trying to say?
Transduction.
Transport. Simple. Okay. It's simple transport. Okay. So we've got it flowing out here, where does it go? What's inside of a myofibril? A sarcomere, okay? So you've got your sarcomere in here. Whole bunch of these inside your myofibril. Calcium floods in it, where's it gonna go to? The actin or the myosin? Calcium goes and attaches on the there's actin and myosin, it's not myosin, where is it? Actin, okay, and we told you there was 3 proteins on the actin, right? You have, which ones are these? Not these ones are your
Oh, that's your
actin. Think of the actin as your pearl necklace. Okay, that's the pearl necklace. On the actin, each one of these has a binding site. Okay, we'll get to that in 1 second, but it's important to understand the binding site is on the actin. Okay? Then we have, I like to call the lounge chair, that the calcium likes to rest on. What is that? Troponin. Okay, so the calcium likes to come in, rest on the lounge chair, and what does it do to tropomyosin? It makes it roll off. Okay, so tropomyosin is the spaghetti looking thing. Troponin is the couch or the chair, however you want to think of it. So when the calcium comes on here, it now takes this and moves it off. And what opens up? What binding site? What do we call it? Myosin binding site on the actin. Okay, so it's the myosin binding site on the actin. Okay.
That's when calcium comes?
When calcium comes and stimulates troponin, it's going to pull, it's going to make this, actually I should write this on the other side so it makes it a little understandable. Okay, so when calcium comes and affects troponin, it deforms it, because these are proteins, right? And we talk about proteins just misshape themselves to have different things happen. Okay, so it's going to misshape itself enough to where it's going to pull this off of the actin, freeing up all of those binding sites. Okay.
So when you say it's gonna take it off, it's gonna take the tropomyosin and the troponin off of the actin,
Yeah, it just kind of moves it out of the way.
and then the myosin attaches.
Exactly. So what you have,
So before it happens, the binding site is binding to the tropomyosin?
it's just covered by the biome, it's covered. It's like this. You see this is a binding site? My hand is the tropomyosin.
Okay.
Okay, I'm covering it. Now, if you had a pigtail and a donkeal tail and I said put it on the hole, would you know where to put it? No, but as soon as the calcium comes in and I move my arm, now you know where to put the tail, right? Pigtail and donkey type thing, okay? So this is what it does. So myosin, I wouldn't put a blind on it so you can see it. So the myosin heads are down here. And as soon as I roll off that binding site, now the head can attach to it. Okay, so that's how the myosin binding site, troponin, and tropomyosin all work together. So right now, myosin doesn't know where to bind to. Now it does.
And calcium makes it.
And calcium, stimulating the troponin, rolls tropomyosin off the myosin binding site.
And the troponin is kind of bound to the tropomyosin.
It's a complex, yeah. the tropomyosin, troponin tropomyosin complex. So they're kind of hooked together, and it pulls them off so that you can see that binding spot, okay? Now you have all of these heads on the myosin, okay? These heads have to be pre-charged before the calcium comes in, okay? So what are they pre-charged with? ATP. So one of these is going to be charged with ATP, then convert it to ADP plus P, and then this is going to be energized. Okay? That energy that it has is going to attract it to that binding site.
That's the myosin
This is the myosin head.
head.
Okay? So the myosin head, you can look on 440, see all those little stubbly things? It's also on 445, the purple heads. Okay, so those purple heads come up and attach. Well, in the process of doing that, it's going to complete its stroke. So it's laying like this, it's going to attach and then go again and pull. When it does that power stroke, what happens to the actin? It slides.
Towards the middle.
Oh, that's a horrible arrow. That's even worse.
Beautiful.
It slides, the actins slide together. Does the actin move or does the myosin move? The actin slides over the myosin. What's connected to the Z line? The actin. We said the Z lines shrink or they come together around the myosin. That's what a contraction is. A muscle contraction is when the Z discs shorten because these little heads, I don't know why I'm having a hard time drawing an arrow tonight. These heads pull it. You have some over here pulling the myosin, pulling the actin both ways.
So when one goes, they go one way.
Yeah. So you have the handles on one side of the myosin, handles on the other side of the myosin, and they both go like this. And they're pulling the actin over the top of it.
So when the myosin connects to the actin, this connects to one site and moves it?
Yeah.
And they kind of pass them off to one another?
Well, it's going to pass them off.
Passes them
It's
down.
going to do its power stroke, which these things are so microscopic that it's not going to go all that far. But they all doing it all at one time, and then they're doing this. So it's getting and pulling and pulling and pulling to where it gets to the point where it can't pull anymore, because it's at its limit of how far the sarcomere can contract.
So they just kind of skim across the top of the myosin heads?
Yeah.
Essentially?
Yes. Okay, so that's called contraction. When those power strokes happen, it's contraction. The ATP that was used to power stroke it is now gone. Now what has to happen? More ATP has to come in and release it so that it can go back to its original spot.
Is that after it's already moved it all the way down?
Yeah.
The chain?
Once this one goes up, moves into this power stroke, more ATP is going to come in and release it. And then it might do it again, depending on how much muscle movement you actually need. If I just do this, only a couple of heads are going to be used. But if I go all the way like this, all the heads are going to be used maybe a couple times to try and get the sarcolemma, or excuse me, the sarcomere as compact as you possibly can. Okay? So you want these to be as close as you can, so you might put a couple heads, do it a couple times.
So ATP to grab one, ATP to let go, ATP to grab another.
Well, no, actually ATP, when ATP comes in the second time and releases it, it actually recocks it and re-energizes it so that it can do it again and go. So you need one to start it, you need one to stop it, but that actually is like cocking the gun again. Like an old 6-shooter, you had to cock it then fire, cock it then fire. That's what you're doing here. You're cocking, energizing, then going. Cocking, energizing, then going. Does that make sense? Once we're done with the muscle movement, what's going to happen? Energy comes in and puts these all, re-energizes, takes them off the myosin. Calcium, what's going to happen to it? It's no longer with troponin, so what's going to happen to tropomyosin?
It's gonna roll back.
It's going to cover those binding sites again. So what do we call that?
Relax.
Relaxation. Okay, so on your myogram, the blue is your latent, which is what that sarcolemma, having the action potential run down the sarcolemma. The red, which is a contraction period, is when the calcium has all these binding sites open. And then the green, the relaxation, is when the tropomyosin rolls back over the binding sites and no more heads can attach and do power strokes. Does that make sense?
So it stops.
Relaxation. Okay. We call that whole thing the cross-bridge cycle.